Okay, so I guess I'll wrap things up here before the Journal Club offering. So I'm Christopher Morris. I'm from Louisiana State University, and joined Midas about a year ago now. And my laboratory is investigating arthropod-borne viruses, and that's what I'm going to give you an overview of here today. So without further ado, quick outline. I'm going to talk to you about what I mean by vector-borne viruses. I said pathogens in the title, but I'm really going to stick to viruses because that's my thing. If I wander too far afield from that, we'll get into trouble. I'm going to talk about the impacts of these emergent pathogens, and then I'll mention some of the specialized components of vector-borne virus transmission cycles and how we account for them, and then give you a little bit of information on what we look at in terms of experimental and theoretical approaches to understanding these complex transmission cycles. So, by vector-borne, I mean arthropod vector. So an arbovirus is an arthropod-borne virus. And critically, it's important to know that what we mean here are viruses that are transmitted between susceptible vertebrate hosts by a hematophagous arthropod following amplification within that arthropod. So this is not a mechanical transmission event. This isn't contaminated mouth parts, contaminated legs, and things like that, and contact-driven transmission. This is a biological event in both the invertebrate and the vertebrate. So I have this gross little cartoon. This is a highly stylized mosquito that's had its poor head lopped off and its legs lopped off. And if this is a wing, its thing wasn't flying too well before we did that to it. But anyway, the point is just to kind of show you what I mean by biological replication. And so what we have here is, with the head removed, of course, this is your blood meal coming in. These black dots are virions, virus particles, of course not drawn to size. So what happens is the virus is imbibed by the arthropod, and it's going to be rapidly digested in the midgut here as the arthropod spills a whole bunch of proteases and enzymes into that blood meal to extract the protein. So very quickly, what has to happen is these viruses have to become associated with midgut epithelium cells and be endocytosed there at that site. If they can do that and replicate within those cells, the next thing the virus needs to be able to do is escape those midgut epithelial cells and get into the hemolymph. Arthropods have an exoskeleton. Their internal organs and tissues are bathed in hemolymph, open circulatory system, and so once the pathogen can get out of the midgut cells, it can essentially go anywhere at once within the arthropod. Where it needs to get, of course, at the end of the day up here is the salivary glands. So once these viruses can get into the salivary glands, the next bite that this hematophagous arthropod takes presumably could deposit virus and create a transmission event. So the vectors that might do this include mosquitoes, ticks, sandflies, and midges. The idea here between this slide and this one here is to show you that there's a world of these things out there. There are alpha viruses, flaviviruses, bunyaviruses, and arboviruses. And if I didn't have to go straight to Russia after this, I would have brought along my arbovirus catalog, which is about 5 inches thick and has over 700 species listed in it. So there are a number of them. Of course, they're not all pathogens, but a number of them are very important human pathogens. And some of those are listed here, including mosquito-borne alpha viruses, including the encephalitides, Eastern, Western, Venezuelan. A very important group of flaviviruses break down into both a tick-borne and a mosquito-borne group, and bunyaviruses, an underappreciated but highly diverse genera. So the impact of arboviruses, so I've told you there's a lot of them, but that's not really enough to drive our interest. So that they cause a significant burden of disease. Some of this is knowable, and so for yellow fever virus, about 900 million people worldwide are at risk of this infection. It causes about 200,000 cases per year and an estimated 30,000 deaths per year. Dengue is the big one right now with its pandemic going on, and the size of the tropical human population. There's 2.5 billion people at risk. That's about 40% of all people on Earth live in dengue endemic areas. It causes at least 100 million cases per year and at least 50,000 deaths per year. As for the other 5+ inches of that book and the pathogens within, most of them are going to be either not diagnosed or categorized as fevers of unknown origin. The diagnostic rationale behind diving further than that doesn't really exist because there is no treatment for these. It's just supportive care, and most people get on with them by themselves. So the impact is likely underestimated, and key thing to note is that many of these are currently labeled as emergent. Emergence gets us to an idea about transmission. So let's just talk a little bit about emergence of arboviruses. We're talking about things that are emerging not necessarily due to our enhanced diagnostic techniques and interest in diagnosing every fever that comes into the hospital, but real things that are changing the transmission cycle and the efficiency of that transmission cycle for these viruses. These can be natural extrinsic effects on the cycle of the virus. They can be man-made effects on the virus ecology, and they can be intrinsic changes to the virus itself. So some examples include, for man-made effects, our increase in rapid global transportation of people, animals, and vectors. We move these pathogens all over the earth with tremendous efficiency and fantastic results sometimes. So some examples I'll talk about later in this talk include things like West Nile virus and chikungunya virus. So by moving things around, we can see one effect here with Aedes aegypti mosquitoes, the vector for dengue and yellow fever. Given a lot of our interest in eliminating this vector, a campaign that took place through the 60s really restricted the geographic range of this vector and drove down infection rates for its transmitted pathogens. That, of course, failed. Reinfestation occurred, and on the heels of reinfestation, we went from essentially no dengue to massive outbreaks now of dengue happening in Central South America. Not just expansion of species, but redistribution of species is occurring as well. So we now have Aedes albopictus, which used to be just here in these red countries. Due to our transportation networks, we now have Aedes albopictus in all of these orange countries as well. And it's the main thing eating me in my backyard in Louisiana. It's also a fairly efficient vector of dengue and a number of other pathogens. So beyond expanding their range and moving them around, one of the things that's important to realize is that a mosquito is not a mosquito, a virus is not a virus. And so the idea here is to show you that these folks, they sampled a whole bunch of Aedes aegypti populations up and down the Mexican western coast there, and they evaluated just how infectable and infectious they might become to a singular strain of dengue. And they found a fairly variable response from population to population. And so this is sort of it, by holding the virus constant, the temperature and environment constant, these folks were able to look at sort of an intrinsic differential susceptibility and transmissibility of these subpopulations of the same mosquito vector. Some other things that are going on to modulate transmission include global warming. This is certainly occurring. The impact on vector range has been talked about a lot in terms of anopheline vectors in Africa. But something closer to viral replication is increased ambient temperatures, and urban temperatures in particular, is going to decrease generation time for a pathogen. When the pathogen's living in the invertebrate, what temperature is it at? Ambient, okay? Ambient temperature. So another cool thing these viruses do is they can jump from ambient straight into our bodies and survive that temperature shift and vice versa. So urbanization is another thing that we've done to increase transmission and encourage the emergence of a lot of these pathogens. So more of us live in cities now than elsewhere. This has led to the emergence of dengue, which is co-adapted with the Aedes aegypti mosquito, which is really built now to live with us. So it's a highly anthropophilic feeder, lives in our houses, actually in our houses, and has come right along with us into our urban lifestyle. This has led to an explosion in dengue cases in the last couple decades. So those are all sort of extrinsic things. When we talk about things that happen to the virus itself, we're thinking about, well, mutations, right? So are mutations occurring that are encouraging more transmissive or less transmissive forms of virus? This can also occur in terms of recombination events, less commonly for these arboviruses, and reassortment of viral RNA segments, again, less commonly, really only in the case of the Bunya viruses that I suggested as one of the genera in the first couple slides there, because it actually has a segmented genome. Despite kind of knowing that these things are happening, we're really only starting to ask what the role is of these sorts of intrinsic changes are on transmission ecology and efficiency. So one example of where this was noticed and may have had a role was West Nile virus. It, of course, was introduced in '99 in New York City, and over the next few years spread all the way across the United States. What happened, let me see if I can play that again. So here's '99 and 2000 and 2001, and then something really changed in 2002 and on. And so this was really interesting what really changed. We didn't expect this. This didn't follow our predictions. We were predicting a lot of north-south movement of virus, and this thing just seemed to steamroll straight across the states and really picked up in terms of transmission when it hit the Great Plains. So what happened there? Well, two things happened, and I can't get into what's causal, but it hit another vector population, so it changed its species, the main transmitting species, but it also picked up some mutations. And so whether or not the mutations were causal for it becoming more transmissible in that vector population is something to consider. But there certainly was a replacement of virus that occurred in 2002. So the original strain that was introduced to North America essentially went extinct. This strain took over and is now really the only one you'll find anymore. This is borne out in phylogeny as well. Chikungunya virus caused another recent outbreak in the Indian Ocean area, and hadn't really seen much chikungunya in a number of decades. This one came out of Africa, got onto Madagascar, eventually to La Reunion Island, and went all around the rim of the Indian Ocean there, and it's continuing to cause infections in Southeast Asia now, where it's replacing has replaced in many areas the indigenous strain of chikungunya. So what happened here? It's again the same sort of story. There was maybe a potential vector change where it went from an Aegypti transmitted cycle to an albopictus cycle, but it also picked up some mutations. And one in particular was singled out as maybe being important for enhanced transmissibility. This was in the envelope protein here. And we did some work, as did others, to look at just how much more or less efficient this made it in the primary vector Aegypti versus the secondary vector albopictus. And it certainly did encourage a fitness increase. So it's certainly something to consider, is whether or not these changes in the virus might actually be causal, might be important in terms of transmission modulation. And that's something we're interested in trying to kind of introduce to transmission models for these viruses. So the transmission cycle for these viruses is quite critical to understand. All of these emergence promoters that I just went through and others will have different impacts on different parts of the transmission cycle. And so kind of understanding the complexity of those cycles is important. So some cycles might only take a few hosts. So others might be quite complex sylvatic cycles where we really don't know the vector or the vertebrate species involved. We'll start with something fairly simple, anthropogenic transmission cycle, where it just goes from human to the vector and back to human. And the simplest one we can talk about here is dengue. We have people going to Aegypti, going to people, and so. So in this case, the human is the vertebrate fuel. We amplify the virus. We infect more mosquitoes, and they give it to our friends and neighbors. Dengue gets a little more complicated than that nowadays, because in many urban areas, it's hyperendemic, and so you'll have more than one serotype. And so there's dengue 1, 2, 3, and 4. In a lot of areas in the tropics, all 4 can be found to be co-circulating in large urban areas. There's a bit of a paradox right now in the literature about whether or not these might interfere with each other or enhance each other. There's some old literature suggesting that during essentially your IgM phase, you might be cross-protected against another dengue infection, but there's a lot of quite recent data to suggest that multiple infections actually can occur. Whether or not this impacts transmission or just disease presentation still has to be kind of teased apart. So when we get into some more beautiful zoonotic transmission cycles here, we can get a whole bunch of these overlapping rings where you have enzootic or sylvatic cycles going on, where we might not know the enzootic host or the enzootic vector. We might not even be aware of what this cycle really is. But at some point, it branches out, maybe causes epizootic transmission, and at some point, gets into something that we care about, usually people or livestock. For tick-borne viruses, this is my only nod to ticks in this talk, they have 4 life stages, 3 of which blood feed, and each of these has a different sort of set of vertebrate hosts it prefers to feed on, which creates for a highly compartmentalized but also very interesting and delicate transmission cycle. So for some of these things, they look so delicate, you don't think they could really sustain themselves, but they tend to do that quite nicely. So transmission drivers. Arbovirus transmission intensity is determined by many factors, including the abundance of these competent vectors, access to sufficient susceptible amplifying reservoir hosts, environmental conditions that are permissive to transmission, and perhaps also the presence of particular viral variants. All of these things have to come together at the right place and time to allow transmission to occur and for them to modulate transmission intensity. So I know you've been waiting for some transmission nomenclature and the appearance of R naught in this. So here's the basic reproductive number, again, being defined for you. Vectorial capacity is the number of secondary infectious bites that arise from a single infected vector. So it's sort of an adaptation of R naught. Within it is this term called vector competence, which I mentioned to you is the intrinsic ability of vector to become infected with, support replication, and then transmit the arbovirus. So how these things get put together is here in R naught, we've got vectorial capacity, and then we use the terms for infectivity of the vector, probability of vector acquiring infection from that person, and the recovery rate. So without accounting for vectorial capacity, R naught for a vector-borne disease will essentially hold the vector constant, okay, and not allow it to have an impact on the basic reproductive number. So when Garrett-Jones incorporated vectorial capacity into R naught, it kind of opened Pandora's box, because now we have all this other information we have about the diversity of vector populations and different susceptibility profiles. So V is not really a very simple term. So to kind of get at it, though, first thing we have to do is break it back out. And so we decompose R naught into the vector and vertebrate sides. And you know, for vectorial capacity, I'm going to go through the main parameters that are currently in it. And for the vertebrate, I'll just mention that really C and R account for essentially the intensity and duration of viremia for arboviruses. Okay. I'm not comfortable with that as a total definition. I think it's a fantastic oversimplification, so that's something we're quite interested in exploring further. So the framework for us here is vectorial capacity. Here it is again in this form. Now let me show you the arbovirus transmission cycle. If we take these terms from vectorial capacity and we lay them over a stylized transmission cycle here, we can see that where essentially in the temporal flow of events here, where these different parameters lie. And so you have the infectivity of a host of vectors, you have the number of bites, infectivity of the vector to the host, duration of infectivity of the host. So let's go through some of these things here. So in vectorial capacity, things that affect the force of transmission and outbreak potential include things like M. M is our vector density, okay? So the number of mosquitoes to the number of humans or animals, depending on your pathogen. So the way to interpret this is that the more mosquitoes per human could mean a greater chance of human coming into contact with an infectious mosquito. So in some cases, this may be more important than others. In a setting where your vertebrate fuel is spread over a wide spatial area, a higher mosquito density might be important in terms of making sure you actually have mosquito-vertebrate contact and the potential for transmission to actually occur. For urban transmission, this might be much less important because for things like dengue, let's say, where you're thinking about maybe a household transmission model, very low levels of, very low densities of mosquito are necessary to perpetuate transmission within a household. So some of the ways we think about this in terms of operational, how do we intercede at any of these points to break transmission down and make it less efficient? So the real control opportunity here in terms of interceding at vector density is to try and reduce the vector population. This is what most mosquito control operations attempt to do by fogging or spraying or larvicide. You're trying to reduce the ratio of vectors to vertebrates to bring down the transmission. You can see it's not a very powerful mathematic term in terms of vectorial capacity, and so you'll have to rely on sort of a linear reduction in M for B. So here A squared is the biting rate, number of bites a vector makes on a human per day. So some vectors preferentially bite humans while others do not. So an example here is maybe, and this isn't anything exact, so don't take this home and tell people about it, but if Culex mosquitoes occasionally feed on people, but otherwise prefer birds, this is going to reduce the fidelity rate and the effective feeding rate. So there's going to be less efficient transmission from bite to bite coming out of that sort of mosquito. If, on the other hand, an Aedes mosquito might be more likely to feed on people, this mosquito is going to be said to have a higher fidelity, and it's going to be more likely to contribute to a human-centric outbreak. It's going to force more of its bites on the important host. So this parameter is squared because when you look at the transmission cycle, we have to consider and account for the bite on the viremic vertebrate, which causes the infection in the invertebrate. And then we have to also get the bite back out of that invertebrate to a naive vertebrate there. So the control opportunity here is to try and reduce the likelihood of the second bite. So this can be done through trying to, let's say, distract bites from being focused on any one vertebrate or the vertebrate of interest to protect. But in general, it's a difficult one to try and conceive of control being effective on. B is vector competence. Again, vector competence is this sort of inherent ability for a vector to acquire infection and be able to transmit it. It was incorporated after vectorial capacity was developed, but it was incorporated because people were seeing, well, we really can't assume that all vector populations have the same intrinsic ability to become infected and to retransmit. So vector competence is highly variable, and it depends on the type of pathogen or the strain of pathogen, as well as the vector species and strain and the subpopulation of that vector. In addition, vector competence is highly influenced by things like temperature and humidity. So again, because the vector is living in an ambient environment, changes in that ambient environment will change its vector competence profile. So it's important to kind of know what your environmental assumptions are and how they're going to influence your beta. So vector competence is dynamic, and one of the things we've recently been working on is looking at how vector competence over time changes within an infected population, following infection, let's say. A lot of the issue in literature there is that if you wait till an ultimate sort of level of infection out maybe 2 weeks post-viremic blood meal imbibing by that vector population, you'll get an ultimate level of vector competence, but that might happen well after that mosquito would be expected to successfully survive in the field. So it's important to account for sort of the rise in infectiousness to a mosquito population through a dynamic process of evaluating data. So finally, we have P to the N. P is the daily survival rate of the vector, and N is the extrinsic incubation period, which is the length of time it takes for the vector to become infectious and be ready to transmit following exposure to the pathogen. So something we'd love to know is the daily rate of the vector P, or the longevity of a vector P. And there are, of course, plenty of estimates of it, but one thing we really don't have any way to do yet is to know how old a mosquito is. And so we have no idea really how old natural mosquito populations are. We can look at mosquito longevity in the laboratory, but I'll tell you that's got nothing to do with how long a mosquito is probably going to live in the field. But we know it's a powerful term because of its position here within this equation. So we know that by decreasing daily survival, we're going to have a pretty solid impact on vectorial capacity values. So a lot of, well, we can't necessarily reduce the extrinsic incubation period for a virus, right, going through a particular population. If we can reduce the survival rate of the vector, that's going to be an important way of reducing transmission potential. So each parameter is important, of course, all by itself, but it's really how they interact in the ultimate formulation here, vectorial capacity, that's critical. And this is something that's hard to isolate and say, this is going to hold true in different places and different times. It's something you really need to appreciate for the moment of a transmission cycle you're interested in or evaluating. So again, to kind of take this stylized transmission cycle and overlay our vectorial capacity parameters here, you can see sort of where at different points some of these parameters might have more or less impact on the overall efficiency of the cycle. So some examples of the transmission cycle that we're looking at to develop a better understanding of these parameters and other parameters that need further characterization because again of the variability inherent in these vector transmission cycles include things we're looking at in terms of vector competence, viremia duration, and intensity. So again, for vector competence, I mentioned the idea of looking at it as a dynamic process over time. But it's also important to realize here that for different strains of dengue, I showed you where one group held the strain of dengue constant and varied the mosquito population. They got different values for beta. Well, here, and others have done this as well, we held the mosquito population constant and we put in different strains of dengue. And you can see that not all virus is the same. So some viruses get out of the midgut and are ready to be transmitted more quickly, more rapidly, as these two curves here are showing. And then others take quite a bit longer. And this is days, but that, a day in a mosquito's life is a pretty dangerous and long thing. Others take quite a bit longer to get out. But we found in this case that even this one that took longer to get out of the midgut and get to the salivary glands for transmission ultimately reached a higher level of potential transmission or infection status in that vector population. So trying to capture strain-specific differences in the pathogen like this, and especially when you're talking about something like hyperendemic dengue transmission, where you have many strains co-circulating, the ability for us to sort of look at the mix of strains that are being transmitted in an area and more accurately account for their contribution to transmission instead of just saying it's dengue 1 or dengue 2, right? Could be very important in terms of getting our predictions correct. In terms of C, or viremia intensity, viremias are inherently dynamic, and transmission is likely to mirror this even at low viremia titers. And so, you have to go through a viremia if you get infected. What's important to realize here is that most of the time, this is held constant. And so we're dealing with the assumption of a threshold value of viremia necessary to create an infected vector. This is an artifact of laboratory study, as far as I'm concerned. And I think right now there's a lot of interest in looking at what's going on in lower level viremias and also in the rise to peak viremia. This is more likely the time when someone who's sick is going to be mobile in the community, maybe infecting vectors of other populations besides the ones in their own homes, and could be critical for reallocation of transmission around a community. Also, there's a lot more attention now to phenomenon of inapparent infections. Most of our data for these viruses, of course, comes from clinical reports. But inapparent infections occur at a very high rate, really, for these infections. And so what is the contribution of them to transmission in these populations? There's an assumption of lower viremia in inapparent infections, and that's why they're not maybe as likely to present with the disease. But that doesn't mean they're not infectious to mosquitoes. And so looking at a lack of threshold for viremia for vector infection. And dynamic acquisition potential, I think, is something that really needs to be done. In terms of recovery, recovery again is something that I think is just really under-investigated in terms of its potential to modulate transmission. And so one quick way of looking at recovery includes looking at, again, strain-specific recovery rate. So if your viremia is going to be different based on the strain of virus you've been infected with as a vertebrate, again, because invertebrates don't ever clear the infection, they have it for life, sorry I didn't say that. If you're going to have a differential recovery rate based on the strain of virus that you received, or perhaps your prior immunologic history, that's going to change the rate at which you might transfer between INR and an SEIR model here. And so again, that's some additional granularity that needs to be explored and I think accounted for. So ultimately, the idea is to, having torn this thing apart, put it back together and reevaluate the impacts of the vector and vertebrate sides on R0 in light of enhanced parameter development. The idea is to hopefully develop operational control options that are going to have the greatest impact on these pathogens. So in review, arboviruses cause infections in both vertebrates and arthropods. Arbovirus emergence events are governed by various external as well as internal influences. Arbovirus transmission cycles can be of highly variable complexity, but are generally rather oversimplified. And mathematical, together with experimental approaches to the study of these pathogens, present excellent opportunities for advanced understanding. So, thank you very much.
Hi, I'm Faiza. I was just curious when you mentioned how there was 4 different types of dengue, and how do you find out which one infected a person? Like, is there a different biochemical pathway, and are there different treatments for the different types?
So for different serotypes of dengue? Yeah, so there's 4 serotypes of dengue, and they cause a lot of trouble for us in terms of diagnosis, because if you've never been infected with dengue before, and you get a dengue infection, serologically, we can tell pretty high confidence what serotype you had. But upon your second infection or your tertiary infection, your serologic response becomes muddied. And so it becomes very difficult to know with any certainty what you had next, okay, or what you had previously if you show up and the first time you come to hospital with disease is really your second infection, let's say. It's tough to get that history back and figure it out. So what I've been trying to do at clinical sites where I work and other colleagues is try and get more direct evidence through viral nucleic acid detection. Since we have good viremias, if you get somebody early enough, you can go ahead and detect and sequence and know exactly what the virus is. But that's not done diagnostically. That's not a routine procedure, and really there's no difference in treatment or anything like that based on which serotype you have. It's more of an epidemiologic concern currently in a population level. So.
Thank you. Do you think it is the advancement of sciences that has enabled us to figure out more about the problem with these viruses, or is it the fact that we are less exposed to these viruses to develop adaptive immune responses against them?
So for people, you mean why are we?
More prone to being infected by these viruses.
Well, I don't know. Yeah, I don't know if we're more prone, or again, this is probably a problem of recognition. As we develop the techniques and interest in using them to really get closer to an etiologic understanding of what's ailing people as they come to clinic and hospital. where we stumble across things that we hadn't been able to notice before because we maybe assumed it was malaria or assumed it was yellow fever or something we already knew about, or we just left it be a fever, So as we get better at doing diagnosis, we find more of these interesting things. I mean, we were doing a study in West Africa like this. We were looking for dengue in a population that was suspected of having a lot of yellow fever. We didn't really think that. We weren't seeing a lot of disease. So we went looking for dengue. We found a lot of dengue, but we found just as much some other weirdo virus that there's only 3 papers on, and no one's really done anything with since the 60s. But it was, it was in acutely ill people, and about a third of the patients there. So, does that make it important? For that population, it does. So, but again, that becomes a problem of recognition, I think, for the most part. So.
Hi, I'm Kanisha. I had 2 questions. The first one was about the map that you showed earlier. I was just kind of curious if you noticed there's a reason why the prevalence of dengue was more in the Africa region as opposed to the U.S. and like the other western side of your map. And also my second question was about, are there any measures that specifically that you're taking for to prevent reoccurrence of it?
And I'm sorry, the second part of your question is about
reoccurrence
of
dengue
in people or environments? Or
yes.
So, well, there's nothing really to prevent you as a person from getting dengue over and over and over again. You'll stop showing symptoms at some point because our assumption is you'll stop showing symptoms at some point because you'll have been permeated by all the different serotypes, and you'll have sufficient antibody to protect. But in terms of that map, that's a highly flawed geopolitical display, I would say, for the most part. And so in Central and South America, we have a much finer resolution on the distribution of vectors and disease than we have in Africa. So that's a surveillance artifact. That's not real. It'd be nice to have an accurate map of Africa, yes.
I have one more question. Is there a specific component of the different stereotypes that you talked about that are conserved amongst the different stereotypes for a person that is having the secondary or tertiary infection? And if so, isn't that a good thing for the immune response?
So you're asking about what's the difference maybe on your second or third infection in dengue?
No, I'm asking, is there anything conserved between the 4 stereotypes?
Well, I mean, they're really very different viruses when you look at them at a genetic level. So phylogenetically, these are different viruses, and they're called dengue because of the disease they cause. But they do have a lot of serologic relatedness, as evidenced by the fact that we can't really tease apart sequential infections by serologic diagnosis. And so this is a big deal in dengue pathogenesis is the idea of antibody-dependent enhancement and whether or not these closely related but not quite good enough to neutralize antibodies might be playing a role in terms of disease progression. There's some very interesting work being done that suggests that there's phenomena of both cross-protection and cross-enhancement between these serotypes. And it's a very interesting time in dengue pathogenesis research, just looking at this data come off and trying to see which way it's going to ultimately point. But there's not a clear understanding or direction yet for that. So.
Is that research coming out of LSU or?
There's a whole bunch of people working on it, and it's quite critical because they're rolling out dengue vaccines now, and they want to make sure that they're doing everything they can not to set people up for enhanced disease, as that's one of the theories. So, but that's happening at a number of different places. So.
Hi, Dr. Lewis. Oh, sorry.
Melissa's not doing her job. No, not you.
It's okay. Thank you. Hi. Earlier, when you were opening your talk, you mentioned that the viruses, they want to be, like, they want to go to the salivary gland. And I thought that was kind of a scary thing to hear, because that would imply
Well, if I personified them
intent.
like that, I didn't mean to give them intent, but you know,
Right.
it's probably fairly Brownian inside the hemolymph, but you know, maybe not purely. So certainly not intended, but yeah. So is that what you meant? I was just
Right.
maybe giving them too much personality.
Exactly,
Okay.
because as
Yeah.
far
No,
as I
there's nothing.
understood,
As far as we know, there's nothing that really targets them to the salivary glands. Certainly, the salivary glands have to have a pretty good receptor motif to be able to take those viruses up, because they do need to get in there. But that's not something that they're really maybe honing in on, because most of the mosquito tissues are going to be able to be found infected.